What is the role of light in seedling growth? Let us dive in to know. Once germination is accomplished, light controls further plant development. We now evaluate these effects and ask whether phytochrome is the only pigment involved. How it acts. Development of monocotyledon as seed germinates. Its coleoptile elongates until the tip breaks through the soil. Between the scutellum and the base of the coleoptile is an internode called the mesocotyl, which elongates greatly after germination in deeply planted seeds. Elongation of the mesocotyl, coleoptile, and leaves is necessary to carry leaves into the light and to establish it near the soil surface. In oats, the growth of the mesocotyl is slowed by 54 hours in continuous red light at an irradiance level of 1/10 to that of moonlight. This effect of light on plants is by far the most sensitive one known. Elongation of the coleoptile must be equal to or exceed that of the leaves it encloses. Otherwise, the leaves would be broken off in the soil. The growth rates of these two organs are coordinated until they reach the soil surface. After exposure to sunlight, the leaves become green and photosynthetic and break through the coleoptile tip. Leaf emergence occurs because light promotes leaf elongation and decreases coleoptile elongation. The leaf growth and inhibition of coleoptile are phytochrome controlled. Showing overall effect of continuous light is to reduce coleoptile elongation, even though for the first few days elongation is promoted. Our conclusion from these results is that light helps attain the growth and maturation of young coleoptile cells. Effects of light on elongating cells reaching maturity are slowed in darkness. More recent studies show that phytochrome is the controlling photoreceptor of light, even though long-term light exposure of high irradiance values is necessary to cause the responses. Maj seedlings grown in light near the soil surface. The mesocotyl had elongated very little, and the first two leaves had emerged from the coleoptile. Each of these leaves was rolled up inside the coleoptile, but when exposed to light they began to unroll. Unrolling of grass leaves is controlled by a typical phytochrome response, low irradiance levels of red promoting and subsequent far-red nullifying the red effect. Low energies of far-red are without effect, and low-energy blue is only slightly promotive. Unrolling is caused by more rapid growth of cells on the concave, to be uppermost, than the convex side. Whether this growth is caused by wall loosening, solute production that decreases the cell's osmotic potential, or both, is unknown. Nevertheless, exogenous gibberellins and, to a lesser extent, cytokinins replace the need for light and phytochrome red. These results suggest that phytochrome causes rolled leaves to form gibberellins or cytokinin, which causes unrolling. It seems safest to conclude that light might induce leaf unrolling by causing the production of gibberellins in concave cells. Development of Dicot seedlings. In dicots, the cotyledones either remain underground by hypogeal development, as in peas, or emerge above ground epigeal, as in beans, radish, and lettuce. In either case, a hook is formed near the stem apex that pushes up through the soil and pulls the fragile young leaves or cotyledones. The hook forms due to unequal growth on the two sides of the hypocotyl or epicotyl in response to ethylene soon after germination. As the hook emerges from the soil, red light acting through PR promotes the opening of the hook. Hook opening results from inhibition by the light of ethylene synthesis in the hook. Accompanying this, light increases leaf blade expansion, petiole elongation, chlorophyll formation, and chloroplast development in leaves. Most of the light promotion of leaf growth, at least in dicots, is caused by heavy infrared. The mevalonic acid pathway completes the formation of chlorophyll A. Some of the chlorophyll A is then converted to chlorophyll B. All of these responses led, within a few hours, to photosynthesis in grass leaves as they break through the coleoptile, or cotyledones or young leaves of dicots. As they break through the soil, while cotyledones of conifers somehow form chlorophyll and become photosynthetic even in darkness, their needles require light for these processes. As photosynthesis begins in leaves and cotyledones, even if food is present, stem elongation is inhibited by light. We now realize that blue, red, and infrared all contribute to this phenomenon, and that cryptochrome and phytochrome are both responsible. The infrared inhibition deserves special attention because it is only observed when these seedlings of other species are young. As seedlings become older, infrared progressively loses its effect.